Hi everyone, my name is Elena and I'm a PhD student at University of Leuven, Belgium. Today I will explore in this presentation the hormonal modulation of reproduction and fertility signaling in porcine wasps. Juvenile hormone is one of the best studied hormones in insects that occupies a central role in development, affecting the metamorphosis, the adult behavior by affecting the sexual behavior, and reproduction, for usually have a gonadotropic role by stimulating the expression of vitellogeny. In social insects, this hormone plays two additional roles, by also affecting the reproductive division of labor between queens that are reproductive individuals and workers that are sterile, and also the age palliatism, affecting, for example, the onset of foraging behavior and other tasks. This hormone has been widely studied in the context of social evolution. The evolution of a sociality is considered one of the major transitions in evolutionary history, which contributed to the very variable spectrum of social behavior and organization observed today in social societies, ranging from solitary to highly social species. Juvenile hormone is thought to be the driven of this evolution, and it is hypothesized that the role of this hormone undergoes several changes during the major transition of sociality. One of the most remarkable differences between primitively and highly social species lies exactly on the fact that, contrary to the highly social ones, in primitively social species there is a lack of morphological cases. It is also known that in some highly social species, for example Vespula vulgaris, there is evidence that the role of juvenile hormone is acting as a gonadotropin by increasing overactivation, but it also controls the production of fertility-linked cuticular hydrocarbons. Although primitively social wasps are key species for being in the functional transition between solitary and highly social, fewer studies have conducted experimental manipulations by measuring several traits simultaneously. So, in this study, we aim to test the hormonal pleiotropy hypothesis across different species of primitively social wasps, specifically to test if reproduction and fertility-linked cuticular hydrocarbons were under joint hormonal control in primitively social species as it was previously observed in highly social ones. The effects of juvenile hormone were then studied in several post-emergence nests of four primitively social wasps: Polistes dominula, Polistes saturn, Miscocytrus metathoracicus, and Miscocytrus castanunga. The same methodology was followed for the four species, however with slightly changes. The nests were collected on the field, brought to the lab and kept in boxes till the emergence of new individuals. The newly emerged individuals were collected and equally assigned to one of the three different treatments that were topically applied in the abdomen: metoprin, that is a juvenile hormone analog; precocin, an anti-juvenile hormone; and acetone, that was used as a control solution. The doses of the treatments were also adapted for each species. Treated individuals were again placed in a box for 10 to 12 days, and at the end of the experiment all the wasps were frozen. Afterwards, the cuticular hydrocarbon profiles of the individuals were collected, and then the ovary status was observed by dissection. All the statistical analyses were conducted using the software R. The difference in ovary activation were analyzed for all the species together using a cumulative linked mixed model that showed that metoprin did not have a significant effect on ovary activation and neither precocin. However, it is possible to observe in the graphic that there is a tendency for precocin always decrease ovary development. In our model, species was not significant. However, the post-hoc pairwise tests show that overall, across the four different species, precocin have indeed a significant effect decreasing ovary development when compared with both acetone and metoprin. The pairwise comparisons between acetone and metoprin were not significantly different. Regarding the chemical analysis, a total of 69 compounds were identified in the cuticle of Polistes dominula. The heat map shows the difference in the relative abundance of the compounds between the treatments, and it's possible to observe that for Polistis luminula, the cuticular hydrocarbon profiles of metoprene and precocin treated individuals are different from each other in the relative abundance of the compounds. The compounds in the lower level of the heat map, here highlighted in red, are expected to be the most linked with an increase in juvenile hormone, since they have a higher relative abundance in the metoprene treatment. The post-hoc tests show that most of the compounds appear to have a difference in the abundance in metoprene treatment compared with the control, but not for the precocin-treated females. Specific cuticular hydrocarbons that we found to be fertility linked and regulated by juvenile hormone in Polistis luminula were the C29, C31, and 7-methyl C33. In Polistis saturn, a total of 41 different compounds were identified in the cuticle. Contrary to Polistis luminula, only a few hydrocarbons differ between treatments, and those compounds are mainly related with metoprene, as is possible to observe here in the second heat map. The specific cuticular hydrocarbons that we found to be fertility linked in Polistis saturn was only the 3-methyl C28. For both Miscocitrus species, all the individuals across the treatment appears to have very similar chemical profiles. For Miscocitrus metathoracicus, we found 41 different compounds, and those that most contributed for the changes in the cuticular profile are shown also in the map. Although any of those was significantly different in the pairwise comparisons between acetone and both metoprene and precocin. Furthermore, some of those compounds, such as 3-methyl C29, C29, and C29-1, showed an increase in abundance in metoprene that is possible to be also linked with the amount of juvenile hormone, although without statistical significance. For Miscocitrus castanonga, we found 47 different compounds in the cuticle, and once more, females from the three treatments expressed a highly chemical similarity. We found no difference for these cuticular hydrocarbons to be linked with ovarian activation, and also no single compound was different in pairwise comparisons. However, similar to Miscocitrus metathoracicus, some compounds, even though significant, show an increase in the relative abundance in metoprene, which is possible to be linked also with the amount of the juvenile hormone. As a take-home message, I would like to highlight the fact that our experiment shows that juvenile hormone acts as a gonadotropin in the 4 study species, with the precocin treatment significantly reducing the overactivation levels. Information that is consistent with the early correlation and experimental evidence in other social wasps, for example, from the tribe Bispin and Epiponine. The cuticular hydrocarbon profile that we further studied to test the hormonal pleiotropy hypothesis showed that the treatments induced subtle shifts in the cuticular hydrocarbon profiles, particularly in Polistis luminula and Polistis saturn, and that some of those compounds were also markers of overreactivation, acting as fertility cues, but not in both species of Miscocitrus. The occurrence of C29 and 3-methyl C29 as juvenile hormone regulated fertility cues is actually interesting, because in the highly social wasp, Vespula vulgaris, those compounds were also among the bioactive worker sterility-inducing queen pheromones, which actually supports the hypothesis that queen pheromones in advanced social wasps evolved from fertility cues in primitive social ones. Finally, in Polistis luminula and Polistis saturn, overreactivation and the production of cuticular hydrocarbons fertility cues are under joint juvenile hormone control. You can check any other information in the article that we published earlier this year, where you can find any other details. And of course, feel free to email me with any questions or doubts. Finally, I would like to thank you to my colleagues here in Belgium, and also to our collaborators and friends in the University of São Paulo, Brazil. Thank you very much for your attention.